I have a great group here that you'll be discussing how we're planning for commercial readiness. And I know this has been a hot topic for a number of years, but I think there's a long way to think about where we are today, and when you're thinking preclinically and early clinical, where do I think about manufacturing? That's something I got to worry about later, right? But really, when do these discussions start, and how do I think adaptively about my process? As we all know, these are highly complex, highly variable processes, and how do we manage that into eventually a commercial process that can be repeatable, robust, and obviously meets the rigor of regulatory bodies? So I'm John Maslowski. I'm the Chief Commercial Officer at Forge and a board member here at ARM. And I'd love to have the panelists each introduce themselves. And really, the important part of this is cell gene therapy, editing, cell therapy, all these different modalities. There's different answers, obviously, for each of them. So our panelists will talk from their experience, obviously, and we'll talk about where their sources are coming from. So I'd like to start with Øystein. You can introduce.
Hello, everybody. I'm Øystein Åmellem, based in Norway. I am work for Thermo Scientific and have been doing that for, 23 years. And I've been involved in product design, innovation around isolation, activation, expansion for all these years. And I've been lucky to be part of the sunrise of T-cell therapies before the car came and sort of saved cell therapy for a while at least. And also been involved in the stem cell area, from adult stem cells to iPSCs now. So that's me. Thanks.
Thanks for having me, John. So I'm Robert Ang. I'm privileged to be president and CEO of Vor Bio. We are a cell therapy company working on two different modalities. One is a genome-engineered hematopoietic stem cell product, which is a next-generation stem cell transplant. And we are also working on CAR T therapies, which are derived from healthy donor T cells. We have the privilege of not just working with a great CDMO, but also building our in-house GMP facility, which opened recently.
Very good. So, Marcus Langtry, I work for Lonza. As you might know, it's a CDMO. We have possibly the oldest CDMO when you look at the cell and gene therapy field. We have 4 sites across 3 continents. From my perspective, I've been working with Lonza for 4 years. I do commercial development for cell therapy. I focus as well in allogeneic, so I take all that technology. And before joining Lonza, I was working for also in cell therapy, a small company called Digenics, that we launched the first allogeneic product approved in Europe. And before that, a bit of big pharma experience, so a bit of a combination, I suppose. And yeah, very glad to be here to have the discussion with you. So.
Thanks. My name is Barbara Sanders. I'm the Chief Technology Officer of VectorY Therapeutics. We're a biotech based in Amsterdam, founded in 2020. So I think of all my co-panelists here, I think we're furthest away from commercial manufacturing as we can get, but still very happy to be part of this panel. We develop AAV-based vectorized antibodies, and before working at VectorY, I was working at Unicure, and before that at Johnson & Johnson Janssen Vaccines. So, thanks.
Excellent. So as you can see, a vast array of experience both on the therapeutic side, CDMO service, so I think we have a great discussion about really when do you start talking about the adaptive process. And I think it's very easy, we commonly hear on panels, to start as early as possible, but as we all know, funding these sort of things can be difficult early. So what are some of the first steps that a preclinical or near IND or entering into clinic phase company should be thinking about, as early as possible? What are some of the most cost-effective ways they should start thinking about a commercial plan? So, Øystein, maybe if you don't mind.
Yeah, I can try and
I know that's a conference by itself, that conversation, but
yeah. No, I think the first thing everybody should do is to really do a proper risk assessment of your process, your supply chain, your raw materials. You might discover that you have a single source component in your process. You might discover that some of the raw materials is not really for prime time commercial use. And you might be surprised to find out that this will take quite significant time to scale up, validate, and get ready with all the documentation and the readiness acquired. So it's all about seeing this picture and then act upon the knowledge you get from that risk assessment and really use people with knowledge to do that risk assessment, take it very seriously. That will be my best advice.
Agreed. Anyone else?
I think from a biotech perspective, so we were recently founded, and even though we're far away from commercial scale, we did think very heavily about this from a start. We really knew that we're going for large disease indications like ALS. We knew that to be able to get the volume and the COGS acceptable at a commercial scale, we had to think now on what will our process be. Indeed, like you said, Øystein, that we wanted to know what scale we're going to be at, what kind of equipment can we already purchase now that is scalable, what kind of bioreactors, what kind of DSP columns, but also a lot in our vector design. What can we do in our vector design to already get high yields, full packaging of our AAV capsids, and investing in a team. We're already invested from the start in process development experts and a lot of analytics so that we understand our product and so can tweak our process from an early start.
So, yeah, I'll say, I mean, I agree with what you just said, Barbara. I think it's the right approach to look at it. Almost like start from the end, see what you're going to need, and then come back. I think the points that you made, Øystein, they're very valid. So I would focus on what is the technology that you're going to be choosing, because that's going to be creating a precedent of what is going to happen next. I will look at the analytics as well. Analytics, I will tackle it very early, because if you don't understand what is your product, what is your key quality attributes, how can you define the best process, how can you optimize, how are you going to know what are your critical process parameters. So these are things that if you don't get right from the very beginning, they'll probably stop you. And I will really focus on those two aspects. And I suppose the partnering is something that you have to think very early as well. I would think about, who is the right partner for a given technology, what are your skills, what would you need from somebody else, and I would be looking at, capacity is one thing, but I would be focusing on capabilities, who has walked the walk that can actually, look at the path that you're going to follow.
Yeah, I'll just add, I definitely affirm Øystein's points. At Vor, we made certain assumptions about our process and product. We discovered a reagent we were using wasn't quite GMP as we thought. We also came across another reagent which we had always assumed we could buy off the shelf because that's what people claimed.
Right.
That was GMP, and you know what? It's not as available as you thought either. Hard lessons learned. Unfortunately, we were able to pivot and particularly dual source, which we eventually thought was very important. So maybe I'll just say a little bit about we chose to use a CDMO at the beginning. Just to be honest, we didn't have a choice. We had raised a Series A, 42 million, which sounds like a lot of money, but certainly not enough to build your own facility. Neither would that have been a wise choice because we didn't even have, preclinical proof of concept at that point. So we chose a CDMO. We vetted several and ended up choosing one that ultimately was a good choice. What we did choose to do, though, is do process development in-house. And that required actually quite an investment of time and resources to really crystallize a process that eventually was robust enough we could tech transfer to the CDMO. And so I guess that's one thing I would urge folk to do, which is think carefully about building that function in-house, because it's never your own until it is. And especially when you have to tech transfer it and teach someone else how to do it, it gets pretty tricky.
Right. And specific, I think Robert too, and maybe Barbara, early on, have you found looking at the patient population, does that impact all how you think about manufacturing and scale, epidemiology, even talking to advocacy groups, like really where patients are and how you expand? Is that something that can be considered early?
Definitely, because indeed these larger indications, you will need a larger volume. So yeah, for ALS, that's definitely the case. And also for the other diseases we're going after, like Alzheimer and Parkinson. It's,
Sure, especially the larger population.
Yeah, so for us it was we knew the patient population was large, so we knew we had to go to the larger scales. But I can imagine indeed for smaller disease areas, you can adapt your strategy according to the smaller number of patients, for sure.
For sure.
Yeah, for us, we stage-gated it. We knew, for example, FDA has never allowed a cell therapy product, without some kind of staggered enrollment. What is that stagger that you expect? You can also figure out generally which sites you're going to use when you forecast that they'll actually come online. What kind of general assumption is reasonable as an enrollment rate per site. And then you can start really mapping out where you think you'll be in terms of, clinical production requirements. So we mapped that out, and then we closely matched that with the CDMO's capabilities and really interrogated, can you do X number per week or X number per month? And that also then helps you think about the equations of, okay, well, when should we expand at the CDMO and pay for that capability? Or when should we also think about dual sourcing with in-house capability?
Right. A lot of factors, obviously, early on, and then, now that we've been given these complex processes, and we're using the term adaptive here today, and I think we've all used different terms, right? It's almost like an if-then statement. If your cell count is this, then your next step is this, right? So what are some of the experiences you had or managed actually adding adaptive to a process? It's a little scary from a quality and a regulatory standpoint for some folks who feel, especially if you have a long-term background, rigidity is key, but clearly not the case when you're dealing with living organisms. So what are some specific advice or examples you have of actually doing that and, quite frankly, doing it real time in some of your earlier stages before you're even moving? Is there some experiences you felt there? So, Øystein, I don't know if you've had some early thoughts there, but...
Yeah, all I can say from a product development, technology development perspective is that we are, we're getting ready for sort of online monitoring, and that will have huge consequences of how we design, for example, a media, a feed system in order to compensate for whatever is lacking in your process at a certain point in time. So I think this will actually change the way we think strategy around product development. I would emphasize, though, the importance of going forward to tool providers and have discussions around your visions and your desire and your thought process around this, because this will actually be a new thing in the industry as we learn how to scale primary cells. It's just a new element. So don't think that everybody has a solution out there. Engage in the conversation and start a dialogue. I think that's very important. I miss that part of the market right now. It's so much secrecy, and we need to open up a little bit to advance the field.
That's very true.
Yeah. I think particularly also when you look at the scale, how large do you want to get for a given indication? You have to look into it. But you have to look into, again, what are you going to start with? But I think we should not forget not only like the size of your DS, but thinking about how it's going to be your downstream, right? So are there the right technologies at the scale that you want to go that are going to help you to go in there? I think, Robert, you were saying about the materials, no, as well. Like, we always assume that the right GMP material is going to be there for the right quantity. I would say the same also for the downstream technologies, for example. Yeah? We always assume that the right downstream technology is going to be there. I do it in a small scale, then I go to a bigger scale, and I'm sure I'll find the technology. Actually, the technology is more designed for autologous a lot of the times and for small scale. When you look at a larger scale, you might find that you have a bit of, some technologies that you have to evaluate, and it's not so easy to find the right GMP solution. So I would also think about it that way.
Yeah, so at least for cell therapies, which we work on, there are obviously your reagents. There's your process, which we'll get back to, but then you have your input material. In our case, the input material comes from healthy donors, but you know what? Not all healthy donors are the same. You might think they resemble each other, but I'll tell you they're vastly different.
Having worked in autologous cells, I know exactly where you're coming from. So.
Exactly, and I would even say that from experience from my colleagues working on allergenic off-the-shelf products, the cell sources for those also vary, and each production run is different. So, one thing that we learned is unless you want a gazillion deviations, you might want to write your manufacturing protocol in a way that gives you a little bit of that flexibility, right? That adaptivity, such that you can totally still meet release spec, but have some flex in the process that allows you to accommodate for those differences.
And in my previous life in the vaccine world, we'd used online USP measurements for viral harvests instead of the typical timing that you do beforehand. So that worked very well, but it did mean a lot of upfront investment in figuring out what measurement, what automatic online measurement can I use in my GMP environment. So you really have to understand your process indeed before you can make such a switch. And especially while you're still building your process and starting to understand it, it's not, I think, not the right time to already infuse the adaptive strategy, but for a later phase,
Yeah, and I would say as well, looking at that, you also have to think who are you going to convince at the end, right? So it's going to be the regulator at the end that has to be convinced that that adaptive strategy has been a success. So again, going back to, do you understand your product, right? Do you have you qualified your product enough? Do you understand it enough? Are you measuring what you should be measuring? Because that is then later the tool that you're going to be using for this adaptability, Of course, you could have compatibility, you could do some part of the clinical trial, but at the end of the day, the more you can justify that those changes that you're doing have not made a difference on your critical quality attributes, that's what is going to matter the most, So think about who's going to be at the end, who's going to be asking the questions, and how are you going to explain it.
That's for sure. The interest in new technologies now, so what's coming up. And I think it's cell and gene, there's different technologies, but I think ultimately the goal is cost reduction, scale up, scale out. I want to ask what excites you now about that part as you're starting to think maybe a little down the road a bit and about scaling out to those populations that you're targeting. But with that, there's always challenges, and obviously the biggest one we talk about is comparability. So when's too late? When is comparability, when is it clinical comparability, and how do we avoid that? In general cases, unless it's absolutely necessary, I think it's very common for every developer to know that the process you use to create your tox material is probably not the same one you're going to have by the time you're in phase 3. I think that's common, or even in commercial. I think we've all know that there's going to be tweaks and turns along the way, but how do we actually introduce some of these things if you come to your pivotal and realize, wow, I really need to go to suspension, or I need to scale up or something. So maybe Marcos, you have an opinion maybe there and think about what you do in Lonza and what excites you there.
So in sorry, can you repeat the last part of the question?
Well, so basically, what technologies now from cell and gene standpoint, because when I think of adaptive manufacturing, what's really sort of the cool thing now that's really everyone's starting to migrate towards? But when you're doing that, how do you think about comparability, and really how does that come into your play, and when is it sort of too late, in a way?
Yeah, so I was thinking about the comparability in the sense, when it's too late. I'm not sure there is a clear answer.
That's right.
When it's too late. So that would be my first approach from the clinical perspective. In terms of the way we would approach it, we would typically, try to set up everything in phase 1 and phase 2, so that you go to your phase 3 with your final product. We would also use scale-down models, so that we would basically, from very beginning, measure and understand. We have techniques like computational fluid dynamics and other things in a bioreactor, for example, world, that you use to understand how to predict what is going to be next. So you have a bit of tools that can help you, again, with the understanding of the process, but you tend to fix it as early as you possibly can, knowing that is not always possible. But I don't think that from a clinical perspective there's a clear go, no go. But maybe you're thinking a different way.
I agree. I think it's always a case-by-case basis, and then you as a team should assess, is this really worth the change? I think that's all. So what do we win, and what is the cost from a comparability perspective? I agree,
Yeah, from my perspective, at Vor, we put patients first. And so if there's a way that we can make a higher quality product for patients or more reliably for patients, then we'll absolutely look at it. I think maybe something for the different vendors out there, we look at it directly kind of relevant to what we're doing. So if you have, say, a new technology that allows more efficient delivery, then my first question will be, okay, do you have it in my cell type of interest? Because you know what, jerkat cells behave differently to pretty much everything else.
Right.
So I'd love to see relevant data that's analogous to exactly what we're doing. And then the next question is that worth the 2 years of investment through an MTA to play around with this versus focusing on something else? I would also say that we kind of think about it very simplistically. So with cells, you can either select them, you can juice them, grow them or make them active or whatever, or you can actually edit them or introduce new information into them. I think there's great vendors working on each of those components. There's some people working on kind of all-in-one instruments.
Right.
But we basically just think about it like, can you juice cells better than other companies? Let's see the data. Can you introduce more information or better information than other approaches? Let's see that data.
Øystein, any thoughts?
Yeah, again, from a tool developer's perspective, due to, of course, the advancement of, let's say, applications into allogeneic, where you foresee a scale-up, we are much more cautious now about trying to find and develop technologies that are truly scalable. For example, a bioreactor system that goes from small to larger volumes with the same technology that is as scalable as possible. And then some of the technologies that are also used in connection with bioreactors needs to be also scalable. Knowing the background of some of these raw materials used in your manufacturing process, they are often comes from an R&D research environment, and then they are transitioned to a cell gene therapy environment, of course, with an added quality. But the scalability, I would argue, is a new topic for some of the tools out there. So we need to harmonize tools and consumables with the scalability of bioreactors. And this will be a new area for us to ensure that we don't push a lot of comparability on the drug developer. And so this is something that you should be aware of, something you should engage in a dialogue around, because it's not intuitive that technologies are scalable. The other perspective that I have been involved in, which is sometimes a pain for drug developers, is that when the tool providers start to do their own comparability studies, because
Right.
we do change manufacturing facilities, we do change from a CMO to in-house, or we change a raw material, and then suddenly we say, hey, we need to do these changes, and unfortunately it involves you. Mm-hmm. So the approach we're taking there is really to be ahead of time and not and engage in a dialogue, plan properly, so that we, on our side, obviously do our homework in what's required for comparability studies, which is then, of course, submitted to the FDA, etc. But then for the other party, the customer, the drug developer, to ensure that there's time and clever thinking around how to do changes. It might involve, seriously, for example, new clinical trials, worst case,
Right.
if there's massive changes. But of course, we try to avoid that. So planning is key here, right? The dialogue is key.
For sure, and I think there's been plenty of companies that come up to this decision and have to think about the state of their business sometimes. And what's your cash? Can you have runway to allow for time for a change? I think what ends up happening, we see a lot in the field, is that if it's a platform company, you sort of get your one approved and get it to your patients and show your platform, and then start making changes in your downstream platforms. I've seen some of that as well. So we see that.
Exactly. I was going to say, you could look at the question from another perspective. No? When should you, rather than when it's too late as well. No? And we know that there's been several studies. I think recently the UCL has done a few. Actually, it changes as we go along, depending on the technology, depending on the actual therapeutic that you're looking at. But it's important to know, is it from a business perspective better to do it after the launch, right? And some people are challenging that. Can you do it? Go to the market fast and then do a change post-approval. Again, I've seen numbers in both directions for different technologies. Some studies that say do it early because you're going to be saving money, and then some other studies would do the opposite. I think from our perspective, it's better the earlier you do it, and as a minimum, think about it. Yeah, be sure that from the get-go you make that decision. You know what is going to be your path, what is the funding you're going to need. Again, what is the partner you're going to choose, and try to do it that way.
For sure. So we talk a lot about, we sort of just went through how do we apply changes in adaptive scale, but it's also as you're starting to approach your BLA, you start looking at the package of what needs to be submitted or you're filing in another part of the world, and you start realizing I have a lot of characterization work I actually need to do, right? So we talk about scaling out, but a lot of talk now about scaling down methods and trying to find comparable small-scale study setups that can actually be submissible and give your critical process parameters an actual evaluation. So what have you experienced on a scale-down model? Is that something, obviously, you may be planning, or is this something that, actually I'm going to do all that characterization right up front? Unfortunately, you don't always have that opportunity because things change, but what are some of your experiences on the scale-down side?
So that's one of the points that we tend to do. As I mentioned before, the scale-down models is definitely from the allogeneic side is something that we believe is quite critical. I mentioned the computational fluid dynamics. There's other methods that we use, but at the end of the day, it's also about building the data. So you actually need to prove it. Yeah, it's not only the theoretical. You need to select the right technologies and then actually test it. Bring real therapeutic data into technologies and see how that model actually works. And you build it by experience. You build it by doing it multiple times.
We've been using a lot of scale down to really small bioreactors, so like 250 mils even, like I don't know, Amber series, for example. Yep. And doing DOE experiments and really using that throughput and then confirming indeed at a larger scale. That's exactly that.
And showing it's representative through same materials construction and media and buffer formulations.
Exactly.
I think that's key. That's something.
And what Øystein also said, the analytics is key. You have to have really good analytics so that you know that what you're seeing is real differences or not, and that you know how to use the scale down data to make some predictive statements about scaling up.
So we're a little far from pivotal studies, but in our IND we did propose a battery of characterization assays that we'd be looking at, and we are faithfully doing those. So I would encourage people, if you're going to tell FDA you're going to do something, you should probably do it.
Probably.
So I think I'll give a shout out to and particularly the work that Mike Lemke and his group did for looking at potency assays.
Absolutely.
So that was across different modalities, but that was a really great interaction dialogue with FDA on that specific topic, and seeing how potency assays can evolve over time during your clinical development. Because we don't really view it as what kind of minimum criteria can we get away with release assays. I think instead it's what are the assays that we think are most faithful to the quality of the product that should be evolving over time as you learn more during your clinical development. And certainly potencies is one of those areas. And so we do really want to invest over time in these characterization assays that may well bubble up into release spec.
Probably the most evolving assay of them all, and I think a challenge for sure, when we're trying to figure out what are the right non-animal study assays to apply in a reasonable way. So So I'd like to take a moment and see if there are any questions from the audience for the panel. See if there's anything that are on your mind, concerned about, or share. We must be doing a great job. That's wonderful. Well, if you have anything...
I'm going to raise a concern.
Yep. Actually, that'd be great. Sure.
The concern is, where does the money come from? Because, you know, money's pretty scarce right now.
Well, that's another one I had jotted down also.
All right, well, sorry to preempt.
No, this is a great time to talk about it.
So I'll tell you a little bit about what we've done. So obviously, we talked a bit about this choice of CDMO to begin with, which made a ton of sense. The choice of building an in-house facility was pegged to our IPO. We were fortunate to raise right at the peak of the market, which has both blessings and curses. But having over 200 million in gross proceeds allowed us to start investing into in-house GMP. Now, what we did, whether it's wise or not, we'll figure that out soon, is that we used part of those IPO proceeds to build an in-house facility right in our office space in Cambridge, Massachusetts. Now, it's pretty modest. It's roughly 9,000 square feet of GMP space with associated quality space as well. But it's right in our building. And so those GMP operators have lunch with the rest of the company every day all together. And the other thing that we did is we negotiated with our landlord such that we could juice the TI, the tenant improvement funds, from our lease, which basically you just plowed straight into our manufacturing facility. Right. And so that actually allows us to have a very modest out-of-pocket expense in building that facility greenfield rather than going to, you know, a standalone facility and trying to do that.
Right.
So I would just going on that, you know, where does the money come from? I will add 2 points. So from a CDMO perspective, I know that Lonza, for example, has developed a new development on demand offer, which basically it is precisely looking at that. It's precisely looking at, let's not go for the multi-million package. Right. Let's go for something that you could, you know, chew. And we see that the companies are struggling with the funding, and that they might want to start, look at my next milestone. This is what I can afford. This is the development I need to prove my next milestone and to get my next tranche of funding. And that's a service that we now provide. So that's the one thing. The other thing I would say is, again, where the money comes from. At the end of the day, you have investors. And I also see, and we have heard these days, that investors are more looking to the value per pound or value per dollar, per euro if you want. And when you look at it from that perspective, it's also interesting to basically think about how do they think, no? Where do they see the value? And a lot of the time, when you invest very early on a commercial path and you can actually prove it, a lot of companies would actually see the value on there, and a lot of investors would actually see the value on there. So, you know, a few years ago, great science would actually get an investment. I think today, a great science with a commercial plan will get an investment.
And some data.
Yeah, and the data. So if you don't have science, if you don't have the data, you know, that's you need that to be on the game. But you also need a commercial plan, and that you have thought about it, and that you can actually prove it to actually get the right investment these days.
Yeah, at VectorY, we're lucky that our investors are also investors that are happy with our commercial mindset from the beginning. So we do run into a lot of investors that don't see that, and they look more at the science. So in that sense, we're lucky. And like you were saying, Robert, at this moment of time, we don't have our own GMP facility and not the finances to build it. So we are also indeed looking at CDMOs for our first clinical productions, and happy that there are CDMOs out there that are working with our budget and our cash constraints. So that is definitely moving in the right direction for biotech companies. Yeah.
Well, it's interesting because leading in, Robert, one of the questions I had thought of is the discussion about automation. And everyone loves automation. It's actually something at Forge we're heavily interested in, want to and invest in. But you bring up the right question. So you have a new board, you have a fresh raise of money in a Series A, and you're going to go to them in time, you're going to spend most of that on automation. So obviously not the right approach. So what is what is the best way to rank and file these things? Is it semi-automation? Is it focusing on analytics, robotics? These are things that will be very helpful when it comes to commercialization and consistency and trying to pull the operator out as much as possible. But what really is, there's a dream path, but what is the realistic path here? So
I can start. I would think that first, if we look at it from an analytical perspective, I would actually look at a qualified method that actually I make sure that I understand what I'm measuring. I think that's the key. Throughput can come possibly a bit later, right? Because you could always scale the analytics as well, and you probably save time on volume. But I would start by saying I want a test that is actually qualified to do this job. From a process perspective, I would be conscious about what happens next. So sometimes you might not go to an automation or go to an open process, and when you realize it, it's too late to change. Right. Or you go into all this compatibility that we talked about, when it's too late, etc. So those things, I would actually encourage people to think about it very early. And if it's related to something that you're going to be struggling to change later on, invest early, even if that costs you a dime.
Yeah, I agree. For automation, we really look at it every time we make a business case. What is it that we're willing to automate, and what will we get out of it? So really looking at more for the analytics high throughput. We'll do it for a qPCR, if you have thousands and thousands of samples. How much throughput will we win in accuracy, and what will it cost us with respect to personnel, expertise, because automation is also a whole new mindset and a new set of skills you have to get in, and of course the equipment itself. So offsetting what you'll get out of it, and indeed whether it's worth changing in your process at that moment in time is what we've done. But for qPCR, we're really looking into it now to automate. Yeah.
And I would add that if you're an innovator or if you're a CDMO, it's also a bit different, no? So a CDMO, I expect an investment in automation for analytics, right?
Exactly.
Because you could do it and you leverage that equipment for many other clients, no? So I would expect my cost of goods to be affected by that as well.
And we ranked ours by risk, really, when we thought about how to approach automation first. And exactly, Barbara, was analytical in PCR, ddPCR, qPCR. High throughput, high volumes, long run times sometimes, and
exactly.
I don't want a person standing there all night sometimes. The other one's filling, too. We felt that with the volumes that we approach. Filling automation going from a manual, even semi-manual, it's quite a jump when you go to full. And if it works well and it fits your modality, we found that there's two of the really valuable points to focus on first. I think from end to end, that may be down the road sometime, but I think the reality is there's always going to be a mix of automation, semi-automation, and unfortunately also some manual. So all the advancements to commercial and technologies and automation, we got to collect a lot of data when we do this. And there's been a lot of, either out of the box or built or, otherwise hybrid systems of how we collect every data point from ERP to analytics and LIMS all the way through environmental monitoring even, and they all are centralized. So what are your thoughts about, this is not something a phase one company needs to think about investing in, but definitely want to start planning a matrix of how we're going to have electronic systems that talk to one another, a CIO to manage it, and how do we do this in a compliant way when the data is submissible? So, and so what are some of your thoughts around these systems? And obviously we're also back to cost again, but, are these a need when you're commercial? And if they are, when's the right time to start thinking about implementation and even inventory, control on that case? So, Øystein, I don't know if you've had experiences on that front.
Yeah, I think this is another emerging area in our industry, and again, I think it's, in terms of automation, I think that's a question of, the level of automation. Automation will come, right? Because we're in the transition phase. Automation will be more standard. 5 years from now, automation is not going to be that much of a topic because we will feel it's natural. It's more a question about, how much do you automate? You can automate your unit operation, the whole process. You can automate your whole plant, for that matter. You can automate all your batch records, right? You can automate multiple level of. So it's a question of when do you actually implement all this level of automation, and they will be available over time, right? So I think that's something that we're looking into, and many are looking into that to really control, because it's so much hands-on work, and you have to take some of that
Programming and
yeah, you have to
training.
take it out. Yes, yeah,
Pulling
and
someone
also
off the
errors in how you operate with the manual systems is too much, I guess.
So you know, I think we obviously collect a lot of data. We have a lot of monitoring as necessary for GMP. I think the important thing is what you do with this, right? You don't collect it just for the sake of collecting and showing it to FDA, say we did our homework, but can we correlate it back to important release or characterization assays that dictate the quality of your product? Could you even correlate it back to clinical outcome if that's possible? You know, and that really depends on what you actually do with the data. What we do at Vor is we feed everything into ChatGPT. No, we don't. I just want to test if you're listening.
Could have done the panel, apparently.
Exactly. We actually have an analytics group that helps us. I mean, this is the same informatics group that does a lot of hardcore science analysis as well, but really helps us analyze the data in terms of just having the highest quality product, right? And really that cross-functional integration between manufacturing, process development, and data analytics is really vital.
For sure.
So I would say, I mean, I personally am a firm believer in automation. I don't see it for like an alternative for commercial. I think it has to be there. When you start looking at the multiple batches that you're going to be doing, it's probably already too late to think about it. But you will need it, both from a manufacturing perspective, so a manufacturing execution system, also the electronic batch records. I think when you get to that commercial stage, you're surely going to need that. At some point, you will realize that you cannot survive without it, almost. I would also agree as well on what are you measuring and why. So if you're not tracing what you're measuring, then it's a bit of a difficult one. And if you don't understand why something is critical, it's a bit of the same thing. So I do think that at the beginning, automation it needs to be in blocks. Now you have to build it up. You need to be again thinking about what type of automation you're going to need at the end, understand it, then let me plan how am I going to build that automation into my process.
Yeah, I think I want to echo the same, especially with respect to the data. There's so much data, and can you make sense out of it? Can you still use the data to say something predictive about your product or your process, and do you understand it, and can you mine it properly? So it's all back to understanding and having the right people to mine that data. Yeah.
So is it possible to collect too much data? I think I've heard a little of that as we were talking. And I think as we approach manufacturing, we obviously collect a lot of data for characterization and consistency early on in our clinical, but as we get to commercial, we hope that number shrinks. So is there too much data to have, and now you own that data, and what do you do with it? So any thoughts around that?
So I would say, again, be sure that you're measuring the right thing.
Right.
And definitely when you go into GMP, definitely when you go into more advanced stages, it's not about measuring more, it's about measuring the right things. Actually, testing is quite expensive when you think about it, and it's, it could be, depending on how you do it, not the best thing that you have in your process, no? It could even become a risk to the process at some point, depends on the sample. So I would think it as, make sure that in your characterization you do enough testing, you understand that testing, you trace that testing, and you actually realize what is actually needed. And I would go with the right level of testing for my GMP. And that is going to be better for the product, better for the cost of goods, and better for the patient at the end, I would think.
Yeah, I agree. I do think there's such thing as too much data, and I've also seen projects where we uncovered data or generated data that we really didn't know what to do with it, and indeed it poses a risk to your program if this data is somehow could pose a risk. So to circumvent that, indeed, develop these methods, develop this data set on not your product, but maybe something that looks like it. Make sure you understand what you're doing. Make sure you understand what the data means, and then once you have that under control, then implement it into your main product and your
process.
No, for sure. And obviously, there's so many, especially with automation, the ability to easily collect data and just have dissolved oxygen probes and CO2 monitoring and everything, it does become quite a ream. And then, I think you can get yourself in a tailspin where you're stuck on the data and you're actually not advancing. So, and then, we've talked a lot about analytics and processing, and I think there's always one topic, I think, with some experience, as I mentioned before, in autologous cells, is in the end, we have to get this to patients, right? So it's the topic of cold chain. And is it necessary for your particular product? And if it is, what are the methods? My experience was I did a 2 to 8 shipment of an autologous cell product, and it's a nightmare. It's very hard to manage all of that. And we actually had a group specifically in our company who just had a big map on a board and tracked packages. So what are some thoughts in different modalities? Like what are we thinking about the right way for delivery? And clinical delivery is one thing at smaller volumes, but once you have a rush and you're doing global shipments of live viral vectors or cells, what are some strategies that we need to start developing early on that front?
I'll say talk at least from an early development perspective. We had to make strategic decisions on starting material. Do you use frozen or fresh starting material? That decision you'll live with for a long time, right?
And you own it all the way, right?
All the way through.
It's a big change, yeah.
All the way through. Then on the tail end, is it terminally frozen or is it not? Is it shipped fresh? That's, again, a decision you'll live with for a very, very long time. And then, of course, you have to still factor in, what about viability, cell loss, because you can't just keep freeze-thawing these forever. So we made certain decisions that we think were prudent, but unfortunately, we didn't have 2 years to test this, right? So we had hypotheses, we tested those hypotheses, they became true, they showed to be true, and we went with that. I would have loved to test other things, but we just didn't have the luxury of time.
Right, for sure.
So I would also say that what are you going to test? So, how stable is your incoming product? How stable is your outgoing product? And that can help you to a certain degree, and the more you understand it, the more flexibility you will have. I don't think there's a magic bullet, though. I don't think you could just say, I'm going to ship, in a completely not cold or refrigerated manner or not frozen manner. It's going to be to a certain degree possible, but only to a certain degree. I think there's going to be challenges, again, both in the incoming tissue and on the final product. Of course, geography also helps. So if you're manufacturing the product and you have the flexibility of accessing different plants in different geographies, definitely for autologous, for example, that is going to be a benefit for you because you're actually minimizing this supply chain issue. But of course, you're not completely eliminating it.
For sure.
Now, I'll just tell a quick story that our first patient that we treated, the collection center was open, had an available slot that we had to live with, and our manufacturer had a particular slot that we had to live with, but product A, need to get from point A to B, and we also had snowstorms to deal with. And we bit the bullet on a private jet.
Oh, I've done that once at a clinical trial.
Yeah.
Oh, yeah. Same reason. Storms in the Midwest. Had no choice, and it's got a shelf life of a few days. So
That's right.
Had to find a route.
And yeah, you got to find a way. And ultimately, this is also something that factored into our decision on in-house manufacturing because, you know what, it's one thing to, work with a collection center, which of course we'll never own or control, or work with a CDMO, which we also will not own, but still somewhat control. But that CDMO probably doesn't necessarily want to start work on a Saturday. Versus I can incentivize my employees to work on a Saturday. And so that is a game changer, at least in the HSC field that we work in.
Sure.
Yeah, and I would add in there, it's also a matter of understanding each other, I would say. I think a CDMO can also be flexible even on that. You need to explain the right reasons why that is important, and there's always an operational model that can actually work. On the other hand, and again from previous experience, I also think that from the clinical side, we need to be flexible. So, sometimes it must be on a Saturday because you have a patient on a Tuesday. But some other times, it's nice to have. Some other times, we could have done it in a different way. We could have done it in a slot on a Monday, and actually the product would come on a Friday. And actually, of course, you could still incentivize people, but if you think about it, actually your cost of goods is going to be beneficial. At the end of the day, somebody needs to pay. So, it's still beneficial that we find operational models that are actually more efficient, both from the operational CDMO side, or if you have your internal manufacturing operations, but also from the clinical side. How are we going to work together? And I think that when you get the clinical and the operational in the same room, we sometimes come with good solutions that can be implemented.
From a viral vector perspective, or the gene therapies I've worked with, they were always deep frozen or frozen, so that was, from a cold chain perspective, doable because the sites always would have a freezer, and we saw with, of course, Moderna's vaccine and Pfizer from the COVID epidemic that these freezers can be made available everywhere, so that's nice. But about 10 years ago, I was also working on an Ebola vaccine of an adenovirus, and this had to be delivered in Central Africa, in really rural places. So how was this done? Once it got off the plane, it went in a little cool box on a motorcycle through the jungle, shaken and everything. Exactly. So what did we do to mitigate everything here is really have incubators at 37 or at least very warm degrees, and really agitation to see, is our product still stable, and can we really deliver these viral vectors? And luckily some of these viral vectors are quite stable, so it's good. Of course you shouldn't do it for months, but yeah, they can have something. But indeed, you have to understand your product.
Wow.
Yeah, exactly, yeah.
Here's your worst case.
So I think we always, all the time, we're going back to understand your product.
Exactly.
Yeah.
Right.
Understand your product, understand what you need, and make sure your product can adhere to what is needed.
Most provocative question might be, is your drug affected by freezing thawing? That's a hard one. That could lead to think about the business model, whether you centralize or you decentralize, and you shorten the wait time by getting your production closer to the patient. It's an area of thoughts that needs to be discussed more.
I think Robert was saying, you have to take those decisions quite early, because then you have to live with them to a certain extent. You could always improve, but again, getting the right choice at the beginning would actually benefit you.
Yeah, so at Vor, we decided to terminally freeze. And there was a little bit of data on this, not much. We kind of went on faith as well as a few experiments. But what was interesting is during COVID, all the stem cell transplants turned into terminally frozen material because no one wanted to collect and then rush it over to the hospital because everyone was, all the precautions and no visitors, et cetera. Overnight, everyone converted to frozen stem cell material. And you know what? Patients got treated, and they did fine. Right. And so that was fortuitous. If it ended up that, all these, graft failures resulted, then we would be in a different place. But we got lucky.
Yeah, for sure.
And if you think about cost, it's the same thing, no? If you take those decisions early enough, yeah, they might be a bit more costly. You might do a bit of development up front, but how much time is going to save you? How much money is going to save you across the cycle?
Yeah.
No, it's true, and I know we're all very COGS conscious, especially when we're starting to have reimbursement thoughts, but no, obviously the quality of product comes first in that particular case. So we have about 5 minutes left, and we're winding down here, so I think I'll just ask one more thing, and I think generally what's really interesting about this topic is we're having it more. So clearly commercialization is now a thing, right? We've had more and more approved products. Arm put up an infographic at the JP Morgan summit earlier this year about all the anticipated approvals. And we have well over a dozen, that are coming up in the cell and gene space, both in the US and in and in across the world, in Europe and across the world. So with that being said, we all have a good problem. If we're talking about commercialization, our data's going well, we're funded, and we're moving forward. So when you wake up at 2 o'clock in the morning, your eyes wide open, what are you worrying about? What is your biggest concern as we're quickly moving this field with a very complex process into a deliverable, consistable manner? So, Øystein.
Yeah, okay. Yeah,
and you have 12 hours to answer that.
No, I think the big elephant in the room is really, I really appreciate all the companies that are going after truly unmet medical needs, because the field needs to move beyond what already has proven to work. It's great to go after CD19 CARs, for example. We know it works now. Those who really dare to take risks, for example, in solid tumors, which is difficult, that's really what I worry about. So I hope that there is courage to go after the difficult ones and really make sure that these fields evolves.
Great. So for us, just think about the funding environment we're in. And I think everyone realizes that preclinical proof of concept doesn't matter anymore in this environment. It has to be clinical proof of concept, and so you need an absolute line of sight how to get there.
Right.
Now, of course, this needs to be in the context that you have a relatively robust, way to manufacture product, as well as you have a relatively well-characterized product that you think could eventually be comparable to what will be your commercial process. There's a ton of caveats in there, right? And there's also always going to be a lot of compromises. But at least in this environment, that line of sight to clinical POC is really what keeps companies alive at this point.
So for me, it would be getting the product to the patient. So that's what wakes me up, if you wish. From the clinical perspective, let's get it right, let's get it done, let's get it on time, and let's treat the patient. That would be my first point. But then, planning for commercial readiness. So that's the other worry, look at how we have done previous interactions with the authorities, how many products we have done that they are commercial already, what we can learn about it, how can we help our clients walking the walk. And at the end of the day, how can we get that product to cross the line so that it's not only clinically effective and you're actually treating the patients, but actually is commercially successful and that we can actually deliver on time for the client.
So I think I have a bit of a combination of you two there. We are going after the harder disease indications in the CNS, like ALS, multifactorial diseases. So why would you wake up at night and think, oh, is it enough that we have the preclinical models aren't fantastic, like they're never 100% predictive, but they also aren't perfect for our disease models. So will it be enough? Will we make a meaningful difference in these patients? And I think definitely in the cell and gene therapy field, we're going for disease modifying, and we have to show a huge benefit to warrant the whole complex manufacturing that we have to do to be able to get to these products. So will we be good enough at this risky time to make a difference to the patients? I think that's...
I agree, absolutely. Well, we're down to 1 minute. So I want to thank all 4 of our panelists. Thank you for your time and your expertise and sharing your stories. I want to thank everyone in the audience. Thank you for listening, and hopefully it was helpful for you. And thank you also for attending ARM conferences. I hope you found this one to be a great one. We love MED, and we love all the other conferences as well. So please, continue to come and talk all these wonderful experts. And I promised you I'd get you to the wine tasting on time, and I did. So all have a great day. Thank you.